Hi, my name is Paul Franks, and I'll be chairing an exciting panel discussion today where we'll cover some of the key lectures on precision medicine at the recent ADA Sci sessions. We'll begin with a welcome from the panelists, each of whom will be introduced. Then we'll start the discussion around each of the different Sci sessions presentations. After each introduction to the lectures that were recorded at Sci sessions, we'll embark on the panel discussion with a little bit of debate, and we'll wrap up with a summary at the end. So I'd like to start by introducing the panel. Mark Anderson, MD, PhD from the University of California in San Francisco. He's a professor in type 1 diabetes and immunology. And we're very happy to have you here, Mark. Thank you for participating.
Pleasure to be here.
We also have Liz Selvin, professor in epidemiology from Johns Hopkins. Thank you for participating, Liz.
Thanks for having me.
And finally, Stephen Kahn, professor in the Division of Metabolism, Endocrinology and Nutrition at the University of Washington in Seattle. Thanks, Stephen, for joining us.
Great to be here, Paul.
And then lastly, I'm Paul Franks. I'm the Scientific Director in Patient Care at the Novo Nordisk Foundation. I'm also a professor at Lund University Diabetes Center in Sweden and an adjunct professor at the Harvard School of Public Health in Boston. So we have essentially 4 different lectures that will be discussed today, one of which is a debate, and that was between myself and Kevin Hall. The selection of these is not an inclusive list of all lectures on precision medicine at the ADA Sci sessions, but it is meant to be a really fairly broad representation around this topic. So we hope that by doing so, and we'll leave it to you to view these lectures online later. They will be freely available, but we hope that that will give you a flavor of what's going on in precision medicine at the moment, and put some things into a little bit more context than perhaps is generally understood. So we'll start with the first lecture, which was delivered by Professor Ewan Pearson from the University of Dundee on pharmacogenetics and the basis of drug response in diabetes. And what Ewan spoke about was fundamentally the role of pharmacogenetics in human diabetes. And he began by juxtaposing the role of genetics in monogenic with polygenic diabetes, or more complex forms of diabetes, like type 1 and type 2 diabetes, and tried to really convey how genetics can help the clinical practitioner. He did so by outlining a 3-point framework, and that included the use of genetics to understand adverse outcomes, to understand treatment response and efficacy, and then also to understand disease etiology. And most of the remainder of his lecture essentially went through supporting evidence for each of these 3 areas. So I'll hand over now to the panel to begin discussing that first lecture.
Well, I'll start, Paul. So, the takeaway for me was that monogenic diabetes, that there's a number of genetic variants that have moderate effect sizes. And that was really interesting to me. It's not a field I know that well, so I was really intrigued by some of the evidence that he presented. And these variants have moderate effects, and they may have some impact on clinical care. So we talked about metformin use and GLP-1 receptor agonists. I think the main point that we all understand is that the genetic architecture of type 2 diabetes polygenic, and the drug response, the evidence, we've only found very small effects, sort of uncommon variants that impact drug response. So I think the real big question for me is that, for changes in the care of type 2 diabetes, we need big effects, right? And so all of the findings, or most of the findings, across the many lectures, is that these are rare, strong effects that we've identified. So, I spend a lot of my time with one foot in the translational space. So the translational piece for me was somewhat distant, right? So I think the big question was, so well, how do we implement this information? How do we go from sort of monogenic, small variant, rare effects, rare variants, big effects to polygenic diabetes, where we need, guidelines need big effects. So that was one of the big takeaways for me from that lecture.
Maybe I can go next, Paul. I found the lecture very interesting, and what Ewan and the group in the UK have been doing, I think, is really making us realize the power of genetics, and not just looking at genetics as a predictor, but using genetics as a means to think about treating people. And some of the insights he gave, which may be new to many people, is the role of genetics even in the adverse effects of drugs, which I thought was very, very interesting. He also, I think, built on stuff that I know Jose Flores touched on, which is these partition polygenic risk scores, which are trying to use an approach that's genetic based, that you can link to a phenotype, which I think is going to help us in the long run pick medications if you know where the phenotype is. But it's really in its infancy. And I think one of the things we have to think about, and I think Mark can talk a lot more to this in type 1 diabetes, is how are we going to find biomarkers that assist us as well in the clinic with genetics? I mean, we're not at the point that genetics is in everybody's electronic medical record. Certainly in the UK, they're getting closer. I think the VA with its Million Vet study is also getting closer to it. But the way healthcare in the US is fragmented between systems, it's going to take some concerted effort to try and bring this all together, that we could be actually seeing specialists in one system and have the genetics with us there that we've been sent from another system. And so I think we've got to think about that. But I will also say, and I'm sure that we can all talk about this a little bit more, as we talk about diversity, equity, and inclusion in the US, and it's a huge focus here, and no doubt, I'm sure it's also coming in Europe. We talk about it, and this talk was at the ADA, must remember that these tools that Ewan and Jose are talking about, that are stuff that we can see in the US, we need to think how this is going to translate to the whole world. And, this is going to be very much the wealthy are going to get access to these sorts of tools earlier than anybody else. And even in the US, there'll be disparities with this. But when we think about what's going to be happening in Africa and parts of Asia, we really need to think what we learn from these approaches, how better and more easily to apply them in countries where healthcare is just not at the same level as we experience in US and in most of Europe.
So that's that's a really important point, Stephen. And I think you're talking there about access to the technologies, I think primarily, but we also have to keep in mind that the vast majority of the data that's being used to design these technologies has come from European ancestry populations, and it isn't a foregone conclusion that they will then transfer well to other ethnic groups, and that's something we'll certainly need to keep an eye on as things progress and develop into clinical practice. Mark, what do you think?
Yeah, well, broadly speaking, since both this lecture and Jose's lecture really get at the issue of how can we use the tool of genetics to dissect a super important problem related to diabetes, and that's the heterogeneity of the disease. And I think that we're all on the same page that both type 2 and type 1 diabetes are heterogeneous diseases, and trying to parse out what your profile of diabetes is of course the goal here. And where does this framework come from, right? So where in clinical medicine has precision medicine really been at the forefront and is already brought to the clinic for clinicians? And I would say without a doubt, it's in our colleagues' clinics in cancer, right? And then in that field, it gets back to what Liz is talking about. So in cancer, they can take the tumor, sequence the genome, find the somatic mutations, which are rare variants with strong effect, strong effects, and that can make a tremendous influence on what the treatment plan is going to be for that patient. And I think that's the kind of thinking and destination we're trying to get at with diabetes. But where I think we stand right now are some big challenges. So I do think that we've made a lot of progress in the rare variants that confer strong biological effects. So a great example of this would be MODY, mature onset diabetes of the young, and with there more than a dozen different forms of MODY at this point that have been identified. And when a MODY mutation is known in an individual, it actually does have an influence on what the treatment plan is going to be for that patient. So there's no doubt in that situation that precision medicine is, but it really is transformative in how you care for the patient. The challenge has been just the broad heterogeneity of adult type 2 diabetes. And then I think the other challenge to bring into the equation that we're going to face gets at what Steve just brought up. People are going to get their whole genome sequenced. The day is coming. These people are going to come to clinic and they're going to hand the clinician their genome and say, what does this mean? And this gets into the details of the problem, because you can, let's just start with the coding part of the human genome and try to dissect out. Somebody has a rare variant in the coding gene. It's been found in a GWAS linkage region in type 2 diabetes. What is the biology of this rare variant, right? And it's very likely that many of the rare variants that are identified in this data have really no biological influence, right? So this is I think one of the major roadblocks for us right now is being able to, be the soothsayer and have, have the Rosetta Stone of what the genetic information actually means in terms of the clinical output. And I think right now what we're starting to see, in this lecture is in pharmacogenetics. You can start picking up some of the low-hanging fruit of variants or genes that do influence metabolism of drugs or having a bad reaction to a drug, but really getting into the precision of, I'm going to choose this particular drug versus this particular drug. I don't think we're ready for prime time yet of using this in clinic.
I agree with you, Mark. I think the sort of the poster child is monogenic forms of diabetes like MODY. And there's sort of a huge expectation that we'll be able to have the same kind of successes with polygenic diabetes, but actually the method is probably going to be different, right? It's not going to be just transferring the approach that's been successfully used in monogenics into complex forms of diabetes. I think there's going to need to be, and there is, people think very hard about how one would do that. And certainly, instead of expecting to find single variants with really strong effects on treatment response, for example, what we may have are more these polygenic risk scores that an accumulation of variants tends to influence the way in which somebody responds to a drug and may mean that another drug is more appropriate. So yeah, so there's lots of a lot more work that needs to be done around complex forms of diabetes. I think we are all saying that. And it's sort of a watch this space at the moment, but it doesn't feel like in terms of predicting treatment response and predicting risk that genetics is necessarily answered today for these more complex forms of diabetes. Although I like what Stephen was saying about the presence of the genome sequence in a patient's record, which could be really valuable for adverse drug responses, which is something else that Ewan touched on, where you might just get a red flag come up if a drug is prescribed because of a genetic contraindication. Yeah.
Well, I just expanded a couple of things I said here. You cannot underestimate like the decoding the biology of what the genetic information means and what a challenge that could be, right? So, one of the fun stories I like to back up to that's very instructive about this is back in the old days, when Francis Collins, our NIH director, was trying to find genes in Mendelian diseases, and they used math with linkage, and they would get to a gene and they would, they'd get to the computer, and Francis would tell the funny story about he would pray that whatever gene came up, it had nothing to do with the immune system. Right? And the reason this is instructive is, let's say we have linkage in a GWAS study to a gene, right? What tissue is this gene operating in? And what time, and then where in the time-space continuum is this gene playing the key role in triggering the ultimate phenotype of type 2 diabetes or type 1 diabetes? And what a huge challenge that is. Many of these genes are not exclusively made in the beta cell or exclusively made in the muscle or the liver. And so, the biology is really tough. And then, layered on top of it is particularly a GWAS, most of the time they find a variant that has real statistical correlation with disease risk. It's in a non-coding region of the genome. And what does that mean? Right? Is it dialing expression up? Is it dialing it down? Is it changing what tissue it's made in? I mean, these are really vexing problems. And so decoding the genome's biology is probably going to be part of the equation ultimately of getting this into clinical utility.
Yeah, so Mark, let me pick up on a couple of things you also
Yeah.
said, and I'm sure Liz found things in there, which is great because we can just chat forever here. But some of these really rare variants, there's actually an NIH initiative on right now called Radiant to try and find novel gene variants for type 2 diabetes, and then to deeply phenotype these people based on other phenotypic characteristics. So if it looks like it's mitochondrial, or to do clamps and all these things. And all of this is really useful because it will help us understand the physiology and pathophysiology related to these gene variants. But I'm still stuck with, if you think back to both, again, Ewan and Jose Flores's talks, if you're going to use clinical phenotypes, and again, I will punt a question in a minute to Liz on this, you're going to use clinical phenotypes. They have to become practical in the clinic to link to this genetic material so that you can say, for example, well, if you've got a high insulin versus a low insulin with this gene, these may be the optimal drugs with what we have now to choose to go with. One can also hope that some of these initiatives like Radiant will actually find gene variants that actually allow us to design drugs. I mean, we think about SGLT2 as a perfect example of a really rare gene variant where these people never get diabetes typically because they pee all their glucose out. And now we're giving everybody a drug to try and get them to pee glucose out, which 20 years ago would have been thought to be malpractice because you'll make people get infections and what will it do to the kidneys? So that's the one side. And then, and I think it even comes back, we're talking about other talks, but, Paul, our chair, talked about nutrition and precision medicine and glucose variability. And so, Liz, I was going to say something to you as to pose, if we're going to start to think about precision medicine, some of the things that relate back to race and ethnicity is things like hemoglobin A1C and these measures that vary between different groups of people, and how we're going to account for this as we go forward to try and make it all work.
Yeah, well, I mean, obviously, I could give a whole lecture on the hemoglobin A1C topic. So I'll resist the urge. I think, so you brought up the tricky issue of race ethnicity. I think the key message I have, or the key thing that I think has not been communicated well in some of the literature, is that it is incredibly important not to use race ethnicity as a proxy for genetics. And I think that happens. I think that's been rampant, right? So people talk about racial or ethnic differences in hemoglobin A1C. What are they talking about, right? So I think it's not clear, right? If you're talking about, race ethnicity is not a precise construct. It's potentially very problematic in the context of medicine. The differences in hemoglobin A1C, for example, they're very small from a clinical standpoint. If it is genetics, right, then it's something that overlaps with race ethnicity potentially, but it is not race ethnicity. Maybe it's a large effect in a very small group or a moderate effect in a broader group. But, so when we talk about race ethnicity, I think we have to be incredibly careful. I'm just using the hemoglobin A1C example because you brought it up, not because it's my favorite molecule, but, so I think it's really important to define things carefully and to separate, if we're really talking about genetics, then we need to talk about genetics. And if we don't know the genetics, we need to declare that we don't know genetics instead of just saying, well, there's racial ethnic differences, we need to do something about it. And concerning to me is there's been lots of calls for race or ethnic specific cut points for certain biomarkers, including hemoglobin A. I think this is incredibly problematic, and, potential to do great harm. So I think I think there's just a lot, and I think a lot of it's a confusion issue, right? There's genetics that hasn't been worked out that we don't understand. And then it's a communications issue in terms of how we're discussing this whole topic. And it's really, and I think we need to be much more careful in how we discuss this. Because I think the health equity issues are substantial, right? And I was really, I thought your comments about globally and the potential of some of this precision medicine discussion and this genetics work to exacerbate inequities, right? Who gets to use genetic information in clinical practice? I think that's one of the sort of things that I kept coming back to in some of these lectures. So I wrestled with that. So Mike Fang at Johns Hopkins just published a paper in New England Journal last month. And, he shows that we're not doing a good job at the basics, right? Only 56% of people with type 2 diabetes are on a statin. right? And only half are achieving a hemoglobin A1C less than 7%. So, you know, I think, and so the access to these new drugs that all of these, you know, lectures are talking about, SGLT2 inhibitors and GLP-1 receptor agonists, right? Only 7% of adults with type 2 diabetes are on one of those drugs, right? So, you know, I mean, we're talking about, focusing on providing specific drugs for people with type 2 diabetes based on genetic architecture, when when access to these drugs is an incredibly pressing problem. So, you know, I kept wrestling with that when I was listening to these lectures in terms of being careful in terms of how we think about the potential for exacerbating health equity when we're talking about implementation of some of these findings.
Right.
Let me just say one thing to you, Liz. You're a little modest when you talked about Mark Fang. You didn't mention you were the senior author on that paper. You should get some credit too. Or maybe you didn't. You wanted the paper known without letting yourself be known. You should be seen as important.
Well, Mike did an incredible job. It's a nice paper.
We're gonna have a very long game day.
Well, I think what Liz is talking about though does bring up a really interesting point too, though, is, you know, and in the clinic, right? We have a lot of work to do. There's so many things that we're not doing efficiently or doing well. But, you know, clearly to me, I think precision medicine is coming. We should be ready for it. And at least, you know, I can comment on in the United States, how we organize medical information in the chart, right? And being able to use it in a forward-thinking way to bring precision medicine back to the patients. I think this is a really interesting area that needs to be developed to make it easy for the clinicians to put in clear phenotyping about the patient that then ultimately could be connected to the genetic information.
So, these are really good points. I'm going to cut in now because we're not going to get through all the discussion. I'll try to sort of say back what I heard, which I think there's there's clearly a role for genetics in precision medicine. Access may be an issue. I do think you can argue that access to some of the imaging technologies is going to be far harder than access to genetic data in developing countries. So it may be actually one of the easier new technologies to implement. But in any case, certainly access.
But Paul, I must stop you because I think there's one important point that still needs to come out here, and it comes back, in fact, to your lecture and the nutrition debate.
Why don't we take it when we get to that?
But it's relevant. I think it's also relevant to all of this. It's very important. We can talk about genetics, and the tools to sequence are pretty standard, and I think you'll get back that information. But I want to take off what Liz talked about at hemoglobin A1C. Try measure hemoglobin A1C. Try measure glucose. Try measure insulin in labs in different cities, and they vary tremendously. So we're going to start to try and phenotype and link it. We've got to be sure we're getting our measurements into order too. That's the important point, I think, that comes out, and I thought is important to raise here too.
Yeah, all right. So it does sound like there's there are a lot of low-hanging fruit that could improve the precision of medicine beyond genetics and other fancy technologies. And I think Andrew Hattersley has actually made that point very eloquently in several papers over the last few years, where he explains that very simple clinical measures can actually be used to stratify drug treatment decisions, which sort of speaks for a somewhat aligned point. So let's go to the next lecture, which was by Jose Flores from the Broad Institute and Harvard Medical School. Jose was speaking in the same session as Ewan Pearson, but he took a different perspective, obviously somewhat different perspective on how genetics can and has been used to improve diabetes prevention and treatment. So he started by explaining very eloquently some of the problems with standards of care for diabetes at this time, and really zeroed in on an important issue, which is that the treatment of diabetes is often done with drugs that tend to deal with glucose, the symptom of diabetes, and not necessarily the processes that lead to diabetes, such as dyslipidemia and liver fat accumulation, obesity, and so forth. And so that was a key point that he made, and he sort of came back to that when he spoke about the partition polygenic risk scores, where he explained how genetics can be used to try to understand the different type of type 2 diabetes that people may have, conditional on these etiological factors that can be genetically characterized. And so that was That was really the first of the 4 ways that he explained that genetics can be used to resolve heterogeneity in the diagnosis of type 2 diabetes. He also talked about, which is sort of an extension of that, stratifying patient groups. And so you might intervene differently in different patient groups depending on their genetic characteristics. He also explained that genetics can be very powerful in implicating new metabolic pathways and molecular targets. And I think probably that's where it's been most intensively used over the last couple of decades, particularly in industry where, and Jose gave some very nice examples of PCK9 inhibitors and so forth that really came out of this very targeted approach with genetics, and explained that actually if you have genetic evidence for the drug target, it doubles the success rate, halves the time from discovery to market, and reduces costs substantially. So it's quite powerful in that sense. And then he talked a bit about causal inference, about, first of all, establishing a causal relationship, not between a genetic factor, but between some other exposure that has a genetic marker for it and an outcome. And this is a method that's called Mendelian randomization. He explained that it's useful for that. It's also useful for determining the direction of effects between an exposure and outcome. And also off-target drug effects might be informed by this approach, where you can study in silico lots of different outcomes that might be associated with the drug exposure. So as is always the case with Jose, did a wonderful lecture that really mapped out these topics very well, in my view. But maybe we can start with Mark's thoughts on Jose's lecture.
Oh, it was a wonderful lecture, and as you said, a great overview of the challenges, the problems, and how genetics can be applied to particularly type 2 diabetes and the drugging of type 2 diabetes. A couple of lessons we'll reiterate. We've already touched on this a little bit. Is the genetic architecture of type 2 diabetes, and what have we learned from GWAS, right? And it really is an interesting story, right? So if you take a Manhattan plot of the entire, right, all the SNPs that are tiled out across the genome, and look at type 2 diabetes, you don't get one huge peak and then a bunch of little peaks or a couple of big peaks. It's very noisy, right? And that's information right there. That confirms that type 2 diabetes is a very heterogeneous group of diseases. And then I think what Jose teased out was showing, underneath all of that data, in fact, you could start grouping people into different categories to some extent, but it also makes it an enormously challenging problem. Now, in terms of the pharmacogenetics, where are the success stories? It's the Mendelian rare variant diseases. PCSK9, SGLT2 came out of super rare patients that had a really interesting phenotype. So the key, of course, was astute clinicians that recognized that something was going on, not only in an individual, but an entire family. And that's where the money's at. And I could tell you right now, I think that's where, as far as industry is concerned and trying to find new targets, that's what they're going for. They are, there's companies like Regeneron that are investing enormously in sequencing whole genomes, looking for rare variants that may have strong clinical effects. And this gets back to this problem I told you about before, was biology. A rare variant, much easier to tease out the biology and show that how that has a relationship to, in this case, metabolism or diabetes. Yeah. So I think that's one thing I just want to underscore.
And one of the points Jose made, which our audience might not all be aware of, is that when you use genetics in drug discovery, what you need to know is not necessarily that mutation has a big effect on the disease. You just need to know that it perturbs that molecular pathway and determine whether that perturbation can be mimicked or reversed with a drug, right? So it's a very different thing. But if you want to use genetics in the prediction of disease, you do need to accumulate large effects, either by putting lots of small effect variants together or finding a few large effect variants. So it's a very different strategy, but it's certainly been, I think it's fair to say, very successful in some cases as a means to discovering drug targets. Liz, what do you reckon?
So, I thought Jose made a very compelling argument that genetics can help. Jose talks so fast. I love it. Each sentence is just like packed with all this information. You're just like glued to the screen, following it. So I always learn a huge amount from Jose. I love this lecture. And I thought he contextualized it in the, when he talked about GWAS and, a lot of the criticisms of GWAS. So I enjoyed his counterpoint discussion and of the important, where he talked about the importance of genetics and the contributions to drug discovery and how, newer whole exome sequencing studies have made important contributions. So I really enjoyed that. And he had this whole discussion about, using drugs that modify glycemia, but the drugs don't necessarily treat the underlying pathophysiology of diabetes. And that's something I've thought a lot about. And, this idea that drugs are treating the symptom and not the underlying disease, the insulin resistance, the insulin secretory defects. So I thought that was sort of a fascinating area of discussion to me, that, when are we lowering glucose with drugs and when are we altering the natural history of diabetes? And I thought, and then he brought in the genetics and talked about, well, how genetics can potentially help and address this question, via studies that really tell us about the natural history. They tell us about the causal pathways, and he talked, this sort of compelling story about the SGLT2 inhibitors falling into that category of a drug that's disease modifying. So to me, that was just a really compelling aspect of his lecture, and I think it's kind of changed the way I think about that area as well.
Stephen.
Yeah, so I think one important thing to highlight from Jose's lecture, which I think we've learned from genetics and phenotyping, both at the level of humans, but also in animal models and clearly in cells, is that I think we always think about a genetic mutation as taking away a function, and that's why you get the disease, right? Or producing something bad. So if you think about the beta cell, if you take away secretion, you get the disease. So if the defect reduces secretion, that gets you the disease. Or if you produce a gene that gives change a gene that creates insulin resistance, you get the disease. But he highlighted the SLC30A8 mutation, which is linked to zinc transport. And we know that zinc is critical for the function of the beta cell secretory granule in terms of processing insulin. And we also know that zinc's related to the insulin molecule and its hexamer structure. Yet what is interesting is that loss of function mutations in this particular gene are enriched in people who do not have diabetes rather than people who have diabetes. In other words, what is zinc transport doing perhaps in excess to create a problem when we think it's so critical to the function of the granule? And I think this again highlights that not everything that where you lose function is a bad thing. Losing function may be a good thing. It also highlights, I think for me, again, another target that pharma could go after to do something different in an area that we don't normally think about. The other thing I think is important to highlight from the point of view of genetics is if you take many of the genes like PPAR gamma and glucokinase, which also links to MODY, these have been shown with really good phenotyping in small populations to be involved in the development of diabetes. But targeting these molecules, PPAR gamma, it's everywhere. Glucokinase, in fact, is a gene that's expressed in many, many tissues. So if we're going to want to target genes like this, and people have tried to be working with glucokinase activators, we have to be certain that we can target them at the tissues that we want. And Mark was touching on this a little bit as well. So I think this is opening up a huge field. I think the pharma companies, as Mark was also saying, are looking for genes which they can target where you've got one gene having effects that you can really work with. But I think we've got to be sure when we're targeting these genes, as pharma will do, that we hopefully can get more precise and be sure that we target them in the tissues that they really need to be addressing, where the defects really are.
And Liz, I'd be interested in, the last part of Jose's talk really was really focused on epidemiology and the concept of using instrumental variables, genotypes as instrumental variables in Mendelian randomization, and using that to try to get away from some of the caveats of observational epidemiology, confounding being a key one. What are your thoughts about the use of genetics in that context?
Yeah, I mean, I think it's very compelling. I think the story and the idea theoretically is very compelling, but you need really strong instruments. Right. So, I mean, and he talked a lot about looking at people at the extremes of the distributions when you're looking at polygenic risk scores and, comparing those people with the very low and the very high, or the high or low genetic load, so to speak, and that can identify phenotypes that may represent pathways that are relevant for therapeutic development. I'm sure, many of the pharmaceutical companies are involved, thinking about this as clustering and figure, trying to parse out the genetics and the physiology that drugs might target. But in the end, I mean, there's only a few examples of those really strong genetic instruments. So, I mean, because that's sort of the key. And so when it all adds up, when it all works, it's really beautiful. But I think the question is, how broadly is that going to happen? How much is that going to, change, right?
I mean, I see your point. I think with if you want a genetic instrument that characterizes an exposure that isn't made up of many gene variants, then you will need large effect sizes for those, or astronomically large populations, right? Which isn't realistic often if you're looking at detailed phenotypes. Conversely, people have done Mendelian randomization analyses with polygenic risk scores, but the challenge there is, and if you go too far, if you add too many gene variants into these scores, you're actually going to be capturing some confounding factors in it, because some of these gene variants, if you go into the thousands of gene variants, will be associated with behavioral traits and things like that. So it isn't straightforward. I completely agree with you, and I think when it works, it can work really well. But it's very challenging to get it to function well in all settings where it would be desired. So Mark, I'm interested in, Jose obviously focused a lot on type 2 diabetes and these partition polygenic risk scores and so forth. What about in type 1 diabetes? Can you see that working there?
Well, at a research level, we definitely are using it right now. And so, the genetic I should back up and just say the genetic architecture of type 1 diabetes certainly is quite different than type 2 diabetes. Genetics are very important in type 1 diabetes. And if you do, what I described before, if you do GWAS and look at the Manhattan plot for how much region confers risk to developing type 1 diabetes, you get one particularly large peak, and that is in the HLA region, right? And the HLA, of course, is the ligand for T cells, and we know that this is underscoring why T cells are important in the disease. But it's a slightly different beast. And so when you look at what now is called the type 1 diabetes genetic risk score, it compounds the HLA type of the patient along with some of the other common variants, which mostly are in immune system related genes. But these other genes confer much less risk than the HLA. So most of what's in a type 1 diabetes genetic risk score these days is really being driven by the HLA. And then if you look at data, so if you take populations of patients, the type 1 diabetes genetic risk score on a per patient basis and saying, oh, you're more likely to be a type 1 than a type 2 is bordering on probably being useless. But if you take a group of patients and look at it, you can parse them out and see as a group of patients where people had autoantibodies, had type 1 diabetes, and you compare them to people had type 2, you can see some shifts. So at a practical level right now, I think one of the notions is to use it as a tool to pluck out people that may have features that suggest they have clinical features that the clinician thinks that might be type 1 diabetes, right? But it really isn't type 1 diabetes. It's MODY, right, or a congenital form of diabetes. That's where right now I think there is some clinical utility to this. And Hattersley from England has published some nice papers on showing that that's where some of the utility is take the type 1 diabetes genetic risk score to identify those individuals that have something different than type 1, right? But again, the measurement on a single patient right now is probably not anywhere near useful, I would say, in the clinic. Ironically, what is useful in the clinic in terms of guessing somebody is on their way to type 1 diabetes? Autoantibodies and their glucose tolerance test, right? High degree of accuracy in predicting whether a patient is on their way to getting type 1 diabetes, right? And so even though we have all this powerful genetic information, it actually turns out the phenotyping is more useful right now.
Yeah. But isn't, Mark, let me just ask you a question here again. You talk about, I mean, the antibodies are really important, But does it not also depend on which antibodies, the number of antibodies, and the age you're at with the number of antibodies? It's very complex, right? It's not just you've got antibodies, you're more at risk.
Right. Well, I guess it depends on what context you're trying to use this, right? To determine does your patient have type 1 versus they have something else, right? You know, even I think in that situation, if they phenotypically look like they have type 1 diabetes, they're thin, they look like they're insulin sensitive, maybe they presented with DKA1 antibody, and you're convinced this patient has type 1 diabetes, right? So I think it gets a little bit gray, but it is true in predictive value. Once you cross the threshold from 1 to 2, that's a big deal. So once you jump to 2 antibodies, your risk of type 1 diabetes starts going up significantly, right? 3, even more, right? And then layered on top of that, an abnormal glucose tolerance test, very, very high chance of developing type 1 diabetes, right? So that's precision medicine. It's just not using genetics, right?
And I'm going to round off this part of the discussion by drilling down on that last point, which is that obviously these first 2 lectures were very much focused on genetics, but precision medicine isn't genetic medicine, right? It should involve other types of data, and some of those types of data may be readily accessible, as we discussed earlier, but it may be that they're combined in different ways, and that will be the method of precision medicine that actually enhances the diagnosis or the treatment decisions, the combination of existing data as well as the inclusion of new data.
Yeah, I just wanted to make one point since we were talking about it. I think this is an important point to bring up is I think a very exciting development in the field of type 1 diabetes that many people know about was a clinical trial that was in the New England Journal about 2 years ago from our colleagues in the TrialNet Consortium that showed that anti-CD3 could prevent type 1 diabetes onset in exactly these kinds of individuals that have multiple autoantibodies and an abnormal glucose tolerance test. And the reason this is important is now there's a treatment that can be connected to predicting whether somebody's on their way to getting type 1 diabetes. Now, the treatment has not been fully approved by the FDA, but it looks like the day is coming in the next year that this is going to be true. And so this will be a precision medicine tool that our colleagues, particularly in pediatrics, will have to start thinking about using.
That sounds like a nice example for precision medicine in type 1. I'll use that, no doubt, in the future.
Yeah. Yeah.
All right. So let's move on. Our 10 minutes per lecture is not quite working out, but we'll move on. So now we're going to talk a little bit about the debate that I had with Kevin Hall around precision nutrition. The debate title that we were given was "Precision Nutrition, Are We There Yet?" It was obviously meant to be polemic and provocative, and I agreed to take the yes argument, even though I could have argued no, but and I think Kevin took the no, even though he might have argued yes in another setting. But it did make, I think, I enjoyed it, at least. I thought it was a good discussion, and what I really tried to do was to to set the frame for what we mean more broadly by precision medicine, but specifically by the role of nutrition in precision prevention. I gave some examples where current lifestyle approaches, the sort of one-size-fits-all lifestyle approaches, or moderately fitted to the individual lifestyle approaches, do not perform outstandingly, even when tightly controlled. And so there's a lot of margin to do better, and we should do better, and therefore opens the door for new approaches, and that might include precision nutrition. And then I gave a couple of examples of large trials that have been conducted over the last 2 or 3 years that provide reasonably good evidence that one can predict an individual's glycemic response to different meals. And that was really the framework of my argument for why it is time for precision nutrition. I did emphasize that I wasn't arguing that it's time to replace sort of standards of care and dietary guidelines with precision nutrition, just that we should be very much focusing on that as a possible way forward. So that was me slinking out a little bit of the hard line of the debate. Kevin, on the other hand, he really drilled down on the technical limitations of continuous glucose monitors. He highlighted that they can be quite noisy, that both in terms of error and bias. And he pointed something out I wasn't aware of, which is that you use the same monitor in the same individual, but you put it in a different part of the body, and you'll get a different estimated blood glucose concentration, and who knows which is the is the right one, the best one to go with. And so you have these challenges that mean that the readout is not precise. And so if you're trying to make precise recommendations, but the readout is a bit fuzzy, that's a significant challenge. He also advocated, and it has another context, for inpatient clinical studies, which is of course what he does at NIH. And we got into the debate part, which about whether that the types of studies that I've been involved in are these free living studies with standardized meals, and of course debated a little bit, one over the other, but ultimately, of course, neither is mutually exclusive, and probably the combination of the two could be combined. The one last point I'll take, which came up in the discussion after we made our pitches, was this point about the noisiness of continuous glucose monitors. And I showed data from the PREDICT trial where we included twins, and we can detect a large genetic component to the postprandial glycemic response based on CGM data, and made the case that if the data was so noisy, you would not detect a genetic component, because that would imply that somebody's genes are influencing the error in a CGM, which is of course nonsensical. So there's definitely signal there, and it can be detected in the context of sort of classical genetic analysis. Anyway, so that's what we debated. So Stephen, you and I have worked quite extensively on the diabetes prevention program over the years. And I used the DPP at the beginning of my part of that debate to explain that, it's not really prevention, it's sort of delay, it's slowing down the progression to diabetes. What are your thoughts about this topic in general, and the two sides to the debate?
Yeah, so Paul, I thought it was really an interesting debate. And of course, somebody like yourself and somebody like Kevin, and it creates, and like I say, you could easily be on either side, which says to me that the debate is needed, but the answers aren't there. And I think the argument to do this as inpatient studies is a good one if you want to try and better understand a little bit of the physiology. But the real world doesn't live inside a clinical research unit in NIH, and it doesn't live inside a clinical research unit with us. And I would also say the diabetes prevention program, as great a study as it's been and continues to be, is a clinical trial where we have research volunteers, and research volunteers don't represent the real world either, yet many of them fail. So I think whatever we learn in these studies, as you're pointing out, has to be taken into the community. And I don't think we can just do them with inpatient assessments unless we want to learn. And it brings me back also, and I think Kevin did this really eloquently when he compared the different machines, right, the glucose meters, that we have to standardize the tools we use and have to standardize the measurements so that, we talk about this all the time. A clinical trial done comparing drug A and drug B, and then you do another clinical trial where you compare drugs C and D, but A and C happen to be from the same class. You can't take the results of one study and compare it really to another one, because the populations are different, the interventions are different, the approaches you've taken are different. And so the same thing holds here. We've got to get ourselves to a point that we can compare apples with apples and close to the same type of apple if we really want to do something with this. And I think both your talks highlighted parts of this, of the elements of being able to predict things, but having to deal with the noise. I do think we have to think about nutrition and as a form of precision therapeutics, if you want to call it that. But then we've also got to think at the populations, and neither of you touch on the fact that if you take populations like youth and you look at type 2 diabetes, is their disease the same? So that what might work for nutrition in adults, we have to then also test these in other populations to make sure they work, and we've got to be able to adapt them. And so I think it's very complex, but I think the debate helped me think that we're getting closer to thinking about these things as feasible, because as a clinician, and I'm sure Mark will tell you the same, he just disappeared for some reason, but Mark will tell you the same from the clinical work he does. We say, well, we should refer these people to nutritionists, but it's so difficult for them too, and they have limited tools to work with that I think learning stuff like you two talked about will be helpful to get us to some point that's valuable.
I hadn't actually seen the paper at at the time of that debate. I saw it like a week later, but you were involved in a nice paper published in Diabetes Care in the spring from Look Ahead that was a secondary analysis, and the subgrouping there suggested that people with poor glycemic control at baseline did particularly poorly when randomized to the intensive lifestyle intervention, which I thought was really interesting. In fact, they were the group that had the highest number of cardiovascular events, and I suppose this is that in itself is a nice it's a nice piece of evidence to suggest that a standard lifestyle intervention, not only may it not work well in some people, it may even cause harm if the preconditions disagree with the nature of the intervention.
Yeah, and let me pick on your talk as well along those lines, where you use the DPP data to show that there's subgroups in there that got no response to lifestyle intervention, and these are people who are at high risk. You think they're being motivated, they want to be in a clinical trial. I think we really do need to understand better what is the difference between that type of group of individuals and the ones who get good responses. And then, of course, the weight issue and lifestyle is something we've talked about for years. You always want to come back to reset yourself back and regulate yourself back. But some people manage. So a lot of that has to do, I think, not only with their motivation, and there are lots of other factors, but perhaps with the nutrition they're applying when they actually get into the program and do this. And so I think there's a lot to be learned here and a lot to be applied.
Yeah. And Mark, so you may or may not have heard Stephen's point. We're interested to hear your perspective as a clinician working with people with type 1 diabetes around nutrition, and whether the sort of stratification of nutritional advice depending or driven by the patient's characteristics is something that is likely to prove useful in the future.
Well, I think intertwined with this is the debate about the real-time sensors, and that certainly the audience is probably even more qualified than I am to comment on this, and just how transformative I think the real-time sensors have been in individualized care and finding out what's going on with the patient. Right? Yes, I mean, you can get down in the weeds about, do the devices have the accuracy to pick up things that we're interested in at a research level? But if you broadly use the data that is now being brought to clinic and look at patterns, it really can be eye-opening on finding things that you did not realize the patient had been doing with their life, with their lifestyle, with their diet, their insulin administration. So I really think that this has been an exciting development for precision diabetes care. Right, like walk through many different examples. Right, so a patient with huge problems with Dawn phenomena that you had never picked up before, right? And the day-to-day variation, the site-to-site variation in using the real-time sensor, yes, there could be problems with it, but the overall patterns really can be quite striking. So I think it's really been an exciting area in the clinic.
So a paper has just come out in Diabetes Care from Erin Sigal's team in Israel, and they were the ones who really broke the sort of ground in terms of this concept of precision nutrition and glycemic control back in, I want to say 2015 or 2016 with the Cell paper. They've just published their clinical trial in people with type 2 diabetes, comparing a machine learning derived nutritional intervention with a Mediterranean diet, and showing that it outperforms the Mediterranean diet in people with type 2 diabetes in terms of time in range on CGM data. So there is accumulating evidence. It'd be interesting to see that transferred into type 1 and see how that performs there. Liz, what are your thoughts?
Yeah, I really enjoyed watching this debate, and it stimulated a lot of thinking. I'm doing a number of studies of continuous glucose monitors right now on people with type 2 diabetes and also with people without diabetes. So I'm thinking a lot about these issues. I mean, a couple of key points for me were nutritional studies in free living human beings is hard. There's profound, measurement issues. I think, when I teach epidemiology to the graduate students here at Hopkins, I talk about confounding, and then I talk about hopeless confounding. So it is important to make a distinction between those two things. And, controlled studies can help, but as has already been discussed, they're going to be limited by sample size. They can't capture, hard outcomes, generalizability, a number of issues. So, I think focusing on improved measurement is critical. And I really enjoyed the discussion of the continuous glucose monitoring in that context. And, one of the takeaways for me is that continuous glucose monitoring is this novel technology. It's very exciting, but it has a number of limitations. It seems to me, and, I've been reading this literature carefully, is that, there's a lot of excitement. And, this technology has, can really revolutionize care, especially for people with type 1 diabetes. And, we're very excited. And in our excitement to adopt this technology, we need to make sure we're also rigorously validating it. And we need to make sure we're also considering all of its weaknesses and not to overlook those. So, it doesn't mean that CGM is not potentially, that it doesn't mean it's not potentially very helpful, very important. But I think in people without diabetes, we need to be quite careful. Right? So glucose is tightly regulated in people without diabetes, right? Our bodies are exquisitely sensitive to glucose excursions. They adapt to differences in dietary intake. And then I think the other thing to me that wasn't discussed in this debate was that continuous glucose monitoring only measures glucose, or interstitial glucose, right? So what about all the other hormones? What about all the other metabolic responses to dietary exposures, right? So, we're measuring glucose because we can measure it, right? So I think an exclusive focus on glucose ignores all the other important nutritional responses. So I think that, and we see that also in the commercialization of this technology. It's like, so now we're focusing on measuring glucose in marathon runners, but glucose is not the only thing that's going to help you run fast. So I think that, just because, we need to do a good job with our measurements, but then we also need to recognize all the things that we're not measuring.
Yeah, and I mean, so in the PREDICT trial, we did actually measure triglycerides and C-peptide as well. But the problem there is that the resolution of the data is pretty low because you're doing this off blood, finger sticks. And although we had the participants do this repeatedly over 2 weeks, that type of data compared with a continuous glucose readout is very poor quality. Interestingly, and we've had lots of discussions with device manufacturers, it is in principle possible to measure other chemicals and metabolites using the CGM type technology. It's just that there isn't the demand to do that yet, but I think perhaps in the future there will be. The other type of technology, and I'm not a technologist, but that I've heard about in this context is pulse oximetry, where you can actually sense glucose using that technology on the with an Apple Watch. Now, whether that will actually ever get to the point where it's sufficiently valid is unclear, but what is clear is that type of technology can potentially be applied to millions and millions and millions of people. So
if we can get it to work. I haven't seen it working very well yet.
No, it's not ready for prime time, but nevertheless, I think technology will march on, and no doubt things will get better.
Paul, can I just say something that's maybe a little old-fashioned here, or maybe it's because I'm old and gray that I can remember some of this, which Liz and you, maybe Mark can remember. I'll put Mark in the older age group here.
Oh, thank you.
You're very welcome. But I think if I recall, you talked about meal composition and how meal composition is so important in driving what your glucose excursion is going to be. And we talk so much more now about ketogenic diets and all these things which are going to change glucose levels. I think we've got to be careful that we don't have people, and we're not talking about diabetes now. I think it was Mark's point about, and maybe Liz's point about people are using this in healthy people. It was Liz's point, healthy people where glucose is incredibly well regulated, and social media allows you to spin whatever you want without any evidence, right? That we don't start to think, well, if we change meal composition, as you showed, I believe, that lowers glucose, that by lowering glucose, we're solving all the problems. It's like you're saying, we've got to measure other things. Giving people high-fat diets forever, I'm not sure is going to be the healthiest thing in other ways. So this is a balancing act. And of course, you can take what is your favorite finding and your favorite belief and push it along. And then you have companies pushing it along. I think we've got to be very careful. At the end of the day, I think nutrition is incredibly important. And being able to use appropriate nutrition and the appropriate people, knowing what outcomes you want and being able to measure them will be valuable. And CGM has certainly helped us a lot in that way, but it should be reserved for what it should be used for, at least based on our current knowledge.
I have to say I applaud Kevin for really pushing this point. I mean, he's done that for quite a while now through many different channels, but that, just because a company tells you a CGM readout is useful doesn't mean to say it is, right? And particularly in healthy populations. And so we mustn't get carried away with the value of that particular readout and ignore everything else. I think Kevin's done a good job of raising people's awareness to that. I'm going to suggest that we move on to the last lecture in this series so that we don't go on too long. And this was really all about kidney disease and the role of precision medicine there, from Jonathan Himmelfarb. I was really touched by the quote that he gave at the beginning from Margaret Hamburg, and this is 10 years old, I think, from the New England Journal, but really demanded that we innovate in our methods, assay standards, and models to shrink the timelines from discovery to clinical application. And I think that was something of a call to arms. I don't know whether that call to arms has been fully and appropriately answered, but it really did sort of, when you have the head of the FDA making these statements, I think it's quite important to sit up and listen. And hopefully the FDA, and of course she's no longer the director, have a philosophy of really trying to scrutinize and facilitate, where appropriate, the approval of these useful technologies. Kevin went on to provide a nice sort of tutorial on how the kidney functions and dysfunctions and why it's very complicated, and really made the point that Jose had made around the diagnostic methods not reflecting the etiology of the disease. And unless you do that, you're unlikely to be able to precisely diagnose and therefore prevent or treat. And then a good chunk of Jonathan's talk was focused on the precision medicine project, which is funded by NIDDK and is a multi-site study, which is quite basic science, but I was very interested to hear about that. Mark, what are your thoughts?
Well, I mean, the first thing I'll bring up is just like what an exciting time it is for being a physician scientist and someone that can translate all of the technology that's at our disposal to clinical medicine, and diabetic kidney disease is yet another area where it's going to benefit from this. I mean, the technology with single cell sequencing of cells for not only the transcripts they're making, but the epigenetic state of the cell, providing something about their identity, even in just normal individuals, is a big deal. Now layer on top of it, you're going to bring in what happens in disease states, right? And so I think where the problems are going to lie with this is when, what are the biopsy material? What are the tissues you're working with? What are the biopsy materials you're working with? Where are you in the time-space continuum of somebody on their way to full-blown kidney failure, right? Where are the events that are happening along the way that really are going to be informative? But the precision that you can have in understanding disease here is really, this is really important work, but certainly not ready for the clinic in any way. That would be that. I think that's my broad brushstroke of it at this point.
And that sort of speaks to the point Liz made earlier today about the need for careful phenotyping, right? One should not get carried away with technology if the phenotyping or the tissues are not appropriate or not properly understood in terms of their origin or timing. So I think that there is a little bit of a disconnect sometimes, I think, between people who have access to, they can do single cell omics, for example, but not necessarily fully, and I'm not suggesting that Jonathan's in that camp, but not necessarily fully understanding the origin of the materials, and so making sure these things are matched up. One of the things I've tried to bang the drum around about a little bit too is in particular in genetic studies, which are often these mega things these days with 1 million+ individuals all pulled together through meta-analysis, is not forgetting about epidemiological principles. So these things that underlie the premise of what you're trying to achieve. And so I think both in that context, but also in the context of doing basic science, you do need to understand the origin of the tissue and the stage of the disease and all those kind of things, which is why having clinician scientists involved in that type of work is really important.
I couldn't agree with you more. And it is funny. It is funny. Steve has already called me out as perhaps an old curmudgeon. And in my field in I consider myself, also an immunologist. My field in immunology, right? Right now, you cannot pick up an issue of Cell Science in Nature, and there isn't a paper in there using this incredible technology phenotyping the peripheral blood of people with severe COVID and what's going on, right? And I hate to tell all of you this, but it seems like many of these studies, there isn't actually a physician involved in the generation of the actual paper. So you have paper with all this complex data and signatures that they see in very sick people that are in the hospital with COVID. And then as a physician, you look at it and you're like, okay, well, what conclusion did I come to here? And it's like, well, they're measuring things that you see in people that are in the ICU. There's nothing COVID specific about it at all. They're missing the controls. They're missing people that are ill with something else that's in the ICU, right? So it does underscore, to me, it underscores a really important role for physicians being involved in this kind of research, right? What do you have the right controls? What is your data actually saying? Because you start having the ability to measure so many different parameters, you get bogged down in the details, in the weeds, without really thinking about, well, what you're seeing here may be epiphenomenon, not phenomenon that's really informing you about how this disease evolved. I'd love to hear what Steve thinks about this.
So I can't agree with you more, Mark, and I take it one step further that, we get all this bioinformatic data, we take tissue and do things in mice. They don't reflect human disease most of the time, right? It's great to study them. We learn a phenomenal amount, but it doesn't translate. And, we know this in your field. Show me a good mouse model of human type 1 diabetes. That's the one side. I think the other thing that I think is important to note that is good for people like Jonathan, who I know really well because we're at the same institution, is he's a nephrologist who can stick a needle in a kidney. I'm interested in the pancreas and what's happening in islets. I can't stick a needle in the pancreas. So to do what the precision medicine project is, or the kidney medicine project, precision medicine project is doing, is laudable. And I think we could do it with the heart. We can do it with the liver. We can do it with a lot of tissues. But when it comes down to something like diabetes and understanding the heterogeneity of the disease and what's happening from the point of view of the islet, and we've talked about the genetics and we can talk about this, it's so much more difficult. And the animal models do not provide a substitute, and we mustn't forget that. So I'm totally with you. We need to think about things from a clinical perspective if we're going to solve human disease. And that's the beauty of having people like Liz and Paul in his old life involved. Liz doesn't do epidemiology on mice.
Correct. I have very, very, very little interest in mice. I want to understand things that are going on in humans. And that's why I have cats. They keep the mice away.
Well, this is sorry, this is, I have to say, just as an immunologist, almost all basic immunologists at some level are mouse physicians. It's just the reality. It's just the reality. I'm sorry, I interrupted you, Liz.
So Stephen, I think those are incredibly important points. I mean, I thought the lecture was very interesting. I mean, the takeaway for me was, the heterogeneity of the phenotypes and diabetes and diabetic kidney disease. I mean, the interesting question of whether, and how treatment strategies can be matched to different phenotypes. I mean, that's sort of the fundamental, theme of the bringing together these different lectures. And I thought, Paul's point was just incredibly important, is that careful phenotyping is essential to answering this question. And I thought, both Jose and Jonathan's talks were really interesting because they discuss how the, emerging evidence that, some medications might be more or less effective depending on the phenotype, but, it's really all about the phenotype, and what we're capturing. And I think maybe going back to how Paul began his debate, right? So he said, he talked about how one size does not fit all, and he talked about shoes. And, maybe I'm showing my bias because I'm a population scientist who focuses on human beings and type 2 diabetes, and I don't primarily focus on genetics. But I do think for some things, one size can work for most people, right? So I don't know, maybe baseball caps, for example. pick your article of clothing, Paul, but baseball caps mostly come in one size. So maybe my curmudgeon statement, to follow up on Mark and Stephen's point, is that for guidelines and for most patients, we need to focus on big effects, right? That's what's really important. And so we need to make sure we're focusing on those things that make major differences in diabetes diagnosis and in diabetes control. And I think, with a lot of this, it's very interesting, but we're still very much at the discovery stage. We don't have those big effects yet that are actually going to change guidelines.
I want to add, sorry, let me add one more thing, Paul, which came from Jonathan's talk, which I think is important because we've been talking about endpoint measurements like glucose, or you might talk about hemoglobin A1C. he highlighted the different pathology, I mean, really bad pathology in a couple instances with normal creatinine clearances. And so we use creatinine clearance or EGFR and say, oh, for example, with the SGLT2 inhibitors, look how they reduce the reduction in EGFR as we follow people over time. But we have no idea what's happening at the level of the kidney. It might be the EGFR. We're sort of lulling ourselves in a falsehood of feeling like we're doing good. So I think that's what we're really going to learn from the sort of work that they're doing in kidney, and we look to other groups to do in other organ systems, where we can actually use the biomarkers we have and relate them back to genetics and to pathology to better understand what's good and bad and how best to intervene. Very
good. I think that might be a good time to wrap this up. We've gone on a bit longer than we've planned, having had lots to discuss. I would like to just close with one thought, which I think it brings together some of the principles of precision medicine and more epidemiological principles, which is that although it's easy and cheap, and I've done it many times to sort of say one size doesn't fit all, it turns out that probably both approaches can be combined. And I'll give you an example of this. So to go back to the precision nutrition debate, in the PREDICT trial, lots and lots of things were measured many times in 1,100 individuals. When you use machine learning algorithms to figure out how you can explain someone's postprandial glycemic response, the most powerful predictor is back of pack information, right? In other words, the information you get on the back of the packet of food, but lots of other things also contribute on top of that. And so, nutritional guidelines are based on back of pack information. We shouldn't be throwing that away. We should be saying, what can we do better on top of that? And I think that applies across the board, actually, with precision medicine. It's not necessarily a case of throwing something out. It's saying, maybe we can still use that, plus a bunch of new things. Okay. Well, I think it's time to wrap up then. Thank you so much for a really stimulating, interesting, and quite funny at times discussion around these really good lectures that were presented at SCI sessions. I know that ADA will be thrilled with this, and I really hope that the audience that's tuned in also have enjoyed this. So thank you.
Thank you.
Thank you.
Thank you.
Well, thank you for those of you who have watched the panel discussion. I found it super interesting and fun. I hope you did too. Some of the key points that came out of it were related to things like health disparity. Will precision medicine narrow the gaps, or will it widen the gaps? We don't know. We have to be careful. We have to be observant, and we have to address these issues as we move forwards, because of course, precision medicine that doesn't serve those most in need will have failed. We talked about the role of genetics. Much of the talk was about genetics, and highlighted the strengths of genetics in the development of drugs and diagnostic tests, particularly in the monogenic forms of diabetes. But we also talked a bit about the limitations of genetics and underscored the need for other types of data, particularly in diagnostic classifications. And sometimes the data is not really the answer, it's the method. We discussed, and I think I enjoyed during the actual debate, and we talked about the debate between myself and Kevin Hall and precision nutrition. Clearly, some of the angles on that are quite esoteric. We talked about the technological readiness of continuous glucose monitors and the need to be cognizant of the error, but the hope that perhaps with new technologies moving forwards, some of that error can be reduced. But also highlighted that all of this has to work in the real world setting, and there are many other factors that influence the food that people eat, and that we shouldn't necessarily cast aside some of our conventional approaches to diet recommendations, because there is value in that. It's just that precision medicine might be able to improve the predictive power of a nutritional choice, for example. And the final lecture was from Jonathan Himmelfarb, who talked about kidney disease and the role of precision medicine. It was quite clearly Jonathan's a clinician scientist, and so he brought an angle of clinical translation to his lecture. But much of it was more about basic science, and he talks about the Kidney Precision Medicine Project and some of the work that they're doing there, and the value of that in developing new ideas around the etiology of kidney disease that might lead to the stratification. And one of the things that resonated in his talk and in the talk given by Jose Flores was really that the diagnostic markers may not be up to scratch. In diabetes, of course, it's primarily a measure of glycemia, either a blood glucose measure or HbA1c, and in kidney disease, it's EGFR. And these are not super precise ways of characterizing the disease because they don't really address the etiological factors that cause the disease. They're more address the symptomology. So overall, we covered a lot of ground. We had some really interesting discussions, and I hope everyone enjoyed the chat. Thank you.